Our next speaker today is Dr. Mei Hong, professor at MIT in Massachusetts. Dr. Hong obtained her bachelor's degree in chemistry with summa cum laude from Mount Holyoke College in 1992 and her PhD from the University of California, Berkeley in 96 with Professor Alex Pines. After a one-year postdoc at MIT, she began her independent career at UMass Amherst before moving to Iowa State in 99. She became a full professor in 2005, held the first John D. Corbett Professorship from 2007 to 2010, and returned to MIT as a full professor in 2014. She has received numerous awards for the creative development and application of solid-state NMR to elucidate the structure, dynamics, and mechanism of action of membrane proteins, amyloid protein, and other biological macromolecules such as plant cell walls. Today, her talk is entitled Binding Site of Hexamethylene Amiloride in the SARS-CoV-2 Envelope Protein from Solid-State NMR. So Mei, if you can go ahead and share your slides, please take us away.
Thank you, Kelly, for the kind introduction, and happy to be here today. So while my slide loads right now, I don't see anything. It takes a few seconds. Okay. So today I want to tell you about my group's recent work in really using many diverse isotopic labeling patterns in solid-state NMR to determine the structure as well as the drug binding site of the SARS-CoV-2 envelope protein. Right. So the SARS-CoV-2 envelope protein, or E protein, is one of the is a member of the family of proteins called viroporins, which are virus ion channels that impact or are important for the virus life cycle, as well as the pathogenicity of many of these viruses to humans. And so this picture shows some important viroporins, including the classical example of influenza A virus M2 proton channel, M2 protein, which my lab has studied for many years, includes some of the HIV proteins such as VPU. And for coronaviruses, this E is the important one. And we know now that SARS-CoV-2 has really only just three structural membrane proteins: the spike, the E, and the M protein. And out of these three, the E envelope protein is the least understood because X-ray crystallography and cryo-EM really can't get to this relatively small membrane protein. So what do these membrane proteins do for the SARS-CoV-2? You all have heard that the spike protein is the one responsible for binding to cell surface receptors, ACE2 receptor, and then causing the virus fusion to the cell membrane. And once the virus is in the cell, this E protein, the new viral proteins and RNAs are synthesized, and then you get these viral components are assembled in a compartment of the cell called ERGIC, endoplasmic reticulum Golgi intermediate compartment, so this region. And here, the M protein is responsible for assembling the various viral RNA and protein components into a new virion, while the E protein is involved in two functions. It mediates virus budding. It makes sure the new virus buds into the ERGIC lumen so that afterwards the new virus can exocytose out of the cell. And then the second function of E is to conduct ions, cations. And specifically, the calcium conducting activity of this protein is associated with the inflammation response of the cell to the virus infection. And this calcium conduction happens because this ERGIC lumen is basically rich in calcium by about 100-fold compared to the amount of calcium in the cytoplasm. So this ion channel activity is important for the virus pathogenic effect. And if you can block it with small molecule drugs, then you have potentially an antiviral inhibitor or drug. And it has been shown that the small molecule hexamethylene amiloride, shown here, has some moderate ability or inhibition to this protein. Obviously, we need better versions, better drugs than this one. But for structural characterization, it's of interest to start with this HMA and its binding site to the protein. So now, to figure out the drug binding site to E, one really first need to know the structure of the E protein itself. And 3 years ago, we determined the first structure of E in lipid bilayers, so phospholipid bilayers that mimic the ERGIC membrane composition. And we did the structure determination at neutral pH and without any calcium or any other cation. And the structure comes out to be this pentameric helical bundle, which is relatively tight with a very narrow sort of water permeated pathway. And you look at this, this is most likely closed state. And there are 5 helices we determined, or later after this study, we confirmed by fluorine NMR that this helical bundle is indeed pentameric. And we know that the pore-facing residues are relatively hydrophobic, rich in leucine, valine, and so on, except for the 2 ends of the transmembrane domain, the N-terminal region and the C-terminal region. These are dotted by polar residues, such as this glutamic acid right in the mouth of the transmembrane domain. There are 3 regularly spaced aromatic residues, phenylalanines, right in the middle of this transmembrane helical bundle. So this is interesting. These phenylalanine residue conformations, we now know, play a pretty important role in regulating the open and closed state of this channel. So this is the knowledge from about 3 years ago. And since then, Joao Medeiros Silva in my group has conducted a very detailed study of the high resolution structure of the open state. So how do we create an open state? What we did was to prepare the proteoliposome sample in a low pH with about 30 millimolar calcium. And this choice of acidic pH and calcium is to mimic the situation of the ergic lumen. Ergic lumen is mildly acidic and is rich in calcium. And under this new sample condition, we can show by NMR, solid state NMR, that the protein becomes much more water accessible. So here you're looking at a 2-dimensional carbon-carbon correlation spectrum that comes from the water magnetization. So it's a water edited 2D experiment. And the intensity, water transferred intensity is much higher under the acidic and high calcium condition, as shown by the greenish, bluish higher contour levels or intensities compared to the neutral pH or closed state. So that right away tells you biophysically this helical bundle is now more open to allow ion passage. Now, for this kind of structure determination of a homo-oligomeric membrane protein, the difficult stage is always how do you get the quaternary structure? How do you get the inter-helical distances as well as helix orientations? And here, isotopic labeling of various kinds is crucial. So if you don't do any special isotopic labeling mixing, just have a uniformly C13 N15 labeled protein, you can measure, through 2D correlation spectra across peaks, but you wouldn't be able to tell what is intramolecular and what's intermolecular. But as soon as you start to do some mixing, for example, you might have 50% of the sample with only C13 labeling, and the other 50% is N15 labeling, then you know your carbon-13 labeled molecules are diluted by 2-fold. And so the chance of seeing an intermolecular carbon-carbon cross peak will be much lower. So most of the cross peaks should come from intramolecular origin. And so that clarifies some aspects of the cross peak assignment. To explicitly look for intermolecular contacts, you really want to take this kind of carbon-13 and N15 label samples and measure nitrogen-carbon cross peaks through various techniques such as NHHC. And those cross peaks can only come from different molecules or inter-helical contacts. So that's the strategy we used. Here we're looking at, in red, a spectrum of a mixed label sample where we dialed in 1,3 C13 label ETM and 2 C13 label ETM. So that's red. And how do we get these? We basically express the protein in with 1,3 glucose or 1,3 glycerol as one carbon source. And then in another sample, it'll be 2 C13 label glucose or glycerol as the carbon source. And that's how you get a mixed carbon label sample. In blue is the spectrum of 1,3 C13 labeled ETM mixed with N15. So this is a control of which contacts come from within each molecule, so intramolecular contacts. And then the second type of intermolecular mixing, isotopic mixing we did, was to mix C13 labeled peptides with fluorine labeled peptides. So, and my lab has developed a number of fluorine-based distance solid state NMR methods in order to measure distances on the order of 1 to 2 nanometers, which are really helpful for solving this kind of homo-oligomeric membrane protein structures. And here we produce this singly fluorinated ETM peptide. This is a 31 residue peptide, so we can synthesize it and just incorporate one fluorinated phenylalanine at one of the 3 phenylalanines that are native to this peptide. And so this is a para position. And now we measure carbon fluorine or proton and proton fluorine distance contacts using the Redor technique. So we detect the carbon-13 labeled protein signals from 50% of the molecules. And we look at how, when you turn on the fluorine pulses, how the intensity of the carbon-13 signals drop as a result of proximity to the fluorine spins. And you can map this mixing time dependent intensity drop and simulate these drops to get quantitative distances. So that's one type of distance constraints. Another type is proton fluorine distance measurement. And this is done under relatively fast magic angle spinning, where you can do proton detected 2D nitrogen proton correlation experiments. And so this would involve a 50% of the molecule that has deuterium labeling, as well as carbon nitrogen. And then we do NH 2D correlation like in solution state. And then again, we then do a second experiment where we turn on the fluorine dephasing pulses so that we can see which amide hydrogens now are dephased by that single fluorine from a neighboring molecule. And in that way, you can map out the amide hydrogen to fluorine distances. So all of these distance constraints, whether it's fluorine-based or whether it's carbon-nitrogen and so on based, led to this open state structure solved at low pH in the presence of calcium. And you can see how these the 5-helix bundle now looks quite a bit different compared to the closed state structure. Now the N-terminal 2/3 of the transmembrane region is really pretty wide and open, looking like a barrel more than a tight helical bundle. While the C-terminal 1/3 of the protein is a little bit tighter. And there are very interesting features to this, you might call alpha helical barrel structure. The N-terminal region is still opened by these glutamic acid residues, but when we looked at it closely, we know now that these glutamic acid residues can adopt two conformations, can either reach out to the next helix and contacting a polar threonine, or can reach up into water where it could then interact or bind protons. And the inter-helical contact pose, we think, is possibly related to the calcium intercalation or calcium binding. The C-terminal tight region, especially at the end of the C-terminal where there is arginine, we think is important for making this channel at the C-terminal end to be polar, and therefore reducing the hydrophobic gate that's in the middle of the transmembrane domain, so that water can actually, and ions can actually be conducted. So we have quite a bit of mechanistic insight from solving the high resolution structure. So now, on the basis of that low pH structure, we really want to know where does the drug bind. Even though hexamethylene amiloride is low affinity, it's a starting point for refining and designing better drugs. And here, this isotopic labeling, different combinations become even more important because we really have to have a handle on the detecting the drug as different from detecting the protein. And so we have nice chemists, collaborators that produced C13 and 15 labeled amiloride, where the squanidinium end is labeled, or we have the hexamethylene ring region fluorinated by different groups of chemists. And then we make sure that the drug labeling is different from the protein labeling, so that when we detect the spectrum, we can choose, we can have a clear case whether we're seeing basically C13 signal from the protein or drug, or fluorine for that matter. We can also fluorinate the protein using our synthetic technology. So here, with a fluorinated drug, you can see how, as a function of temperature, how the fluorine line shape broadens as you go down in temperature. And you can see at high temperature, there's a single chemical shift. As you go down, there's two. We think that two peaks has to do with these two fluorines on this conformationally flexible hexamethylene ring. So that's one piece of information. And now comes the question of finding out where does the HMA bind relative to the protein. And so here's the first piece of data where we combined the C13M15 labeled drug with a fluorinated peptide. And here we introduced the CF3 trifluoromethylfluorinated phenylalanine into the N-terminal region of the peptide. And we detect the carbon-13 signal of the squalidinium moiety of HMA. And we want to see how that carbon-13 signal of the drug is affected by the fluorinated protein as a way of saying, okay, where's, how far these two are. It turns out the more important information is actually the binding stoichiometry, because when you prepare 5:2 ratio of protein monomer to drug, and you detect the carbon-13 signal of that two copies of drug, you find that the redoor dephasing due to the fluorinated protein comes down to about half. And what that means is that only one out of the two drugs is close enough to any of these peptides. If you have a sample with 5:5 monomer protein to drug ratio, we find that the carbon-13 signal of the drug decays to about 80%. In other words, only 1 out of 5 drugs is in close contact with the protein. So both these data indicate that for the 5-helix bundle, for each 5-helix bundle, on average, there's only one drug. So that's an interesting stoichiometry. Now you might think, well, okay, one drug perhaps is sitting right in the middle of the channel. That will be a direct pore occlusion model. And I'll show you the data that indicates this is not true. But here first is another combination of isotopic labeling where we have C13 and M15 labeled drug, and nitrogen appears only in the drug. And we have a carbon-13 labeled protein, so the protein doesn't have any nitrogen. And we do carbon-nitrogen dipolar coupling measurement. And here the protein signal C alpha region can be dephased only by the M15 labeled drug. And we see that indeed we have dephasing. So that means these two molecules are in close contact. And now let me switch to the last combination of isotopic labeling, where we have fluorinated, now HMA, combined with M15 and proton amide hydrogen labeled ETM. We do our proton fluorine reador experiment as detected by the protein-proton nitrogen 2D correlation spectrum, and you can see which amide hydrogens are in close proximity to the fluorinated drug. And when you do this, okay, now we're detecting the protein, you see that only about 80% or so, 20% or so of the amide protons are actually dephased by the fluorine. Okay, so out of the 5 helices, on average, only 1 is affected by the protein, by the drug. So what this means is that this one copy of the drug that's in contact with 5 helices of the bundle, that one drug is not centrally located in the pore. Rather, it is binding from the lipid side. So when we put all of our distance constraints into the Haddock software, we can see, so it's distance constraint docking, we can see various poses that show the HMA molecule, which has this long aromatic, planar side, to interact with the 5-helix bundle in poses that are all coming from the lipid side, whether it's bridging 2 helices or perhaps parallel to the 2 transmembrane helices, or perhaps tangential to the 5-helix bundle surface, or perhaps intercalating into 2 neighboring helices. They generally are coming from the lipid side. The overall orientation can be, as I said, parallel to the long axis of the transmembrane helices, or it can be perpendicular coming in this way. But they're all pretty close to the central portion of the protein where you have these aromatic phenylalanines. And so I didn't tell you that these phenylalanines are important for gating. I mentioned it briefly, but we think that the fundamental mechanism of this inhibition has to do with the HMA interfering with the gating mechanism of phenylalanine so that it can keep the channel in the closed state, preventing ion conduction. So with that, I would like to stop and thank Noah, who did all of the drug work, and Joao, who's the person who solved the open state structure of ETM. And collaborators here, thank you for your attention.
Awesome. Thank you so much, Mei, for such a wonderful talk. We did have one question that came in through the Q&A. Why would nature make modification to slow down the process?
Why would nature make slow down what? Ion conduction? No?
I believe so. It came in the earlier part of your presentation.
So nature did the other thing. In the acidic and calcium-rich ergoglumen, that condition will propel the channel to open. And so once the channel is open, then the virus can, have its effect to trigger the inflammasome. And so I would turn this around and say the virus has evolved its amino acid sequence so that it can respond to the condition of the acidic and calcium-rich environment and open the channel. So that's how the virus works, uses E for its purpose.
Wonderful. Thank you, Mei. And I think due to timing, we're going to have to move on to our next speaker. We did have one more question that just came in. So May, if you're able to answer it, you should be able to in the Q&A section.
Okay, a water edited 2D carbon-carbon correlation experiment. So what we do here is to transfer water magnetization specifically to the protein protons and then detect it through the protein carbon-13 signals. And in this way, we can find out which residues in the protein are most water accessible and which are least water accessible. And this has been very useful for defining the structural topology of various proteins, whether membrane proteins or amyloid, but particularly useful for membrane proteins that have a water-permeated pore versus the lipid-facing side. Yeah. And this is an experiment that is really special to solid state. I think in solution, it wouldn't make sense to do such a thing, or it couldn't be done. Yeah. NMR more than obtained for acquire. Okay, that's a question. I have to answer that. So what is specific about NMR? So virus ion channels are basically impossible to study by cryo-EM because they are small membrane proteins, too small for cryo-EM, and they have very little water-soluble portion for crystallographers to use to make crystals. So they're perfect for solid state, for NMR, for solid state NMR. Yeah, and you can also look at them in the lipid bilayers. These small ion channels, if you put them into detergents, they tend to then adopt some different conformation. Anyways, it's could be artifact. Yeah.
Wonderful. Thank you, May.